Okay, Neil, now is the science lesson that I've been waiting to give to you. Now, I'm not a scientist, first of all. However, I've spent the last 18 years interviewing scientists, not on the payroll of the biotech industry, but who have genuine concerns, even evidence showing that GMOs are unsafe. So when I write books or make movies, I show the script and the manuscripts to the scientists to make sure that I've got it right. So I'm pretty confident that what I'm saying comes from scientific authority, and I think when you look at the details behind it, you'll agree. The process of genetic engineering is not like sexual reproduction. Let's say you want to create a corn plant that produces its own toxic insecticide. So first you find a natural toxic insecticide like Bt from Bacillus thuringiensis in the soil bacteria world. You take the gene out that produces the toxin that breaks open the stomach of insects to kill them, and you make millions of copies of that gene. Then you put it into a gene gun, and you shoot that gun into a plate of millions of cells, and then you identify which one of those genes got into which of those cells, and then you clone those cells into a plant, and now every cell of those corn plants has a gene-sized spray bottle that can kill insects. Now, the process of genetic engineering, whether it's done by gene gun or bacterial infection, and followed by cloning, creates massive collateral damage in the DNA. 2 to 4% of the DNA is different in the genetically engineered plant. The number of mutations is vastly greater than that which occurs during sexual reproduction. So right there, you have a difference that's profound and that can lead to danger and lead to unpredicted side effects. For example, in Monsanto's most popular corn variety, Mon 810, it produces the Bt toxin, they found that a gene that is normally silent in corn was accidentally turned on during the process of genetic engineering. And this gene creates an allergen called gamazine. So people might get allergic reactions to Monsanto's corn. They might even die from allergic reactions from Monsanto's corn to an allergen that is part of the process of genetic engineering, not labeled, and part of the side effects. In fact, there were 43 different proteins that were vastly upregulated or downregulated or switched off or switched on. And some proteins, which is a big concern for the authors of this study, had completely different shapes. They were truncated. And you know, Neil, that when you take a protein and you change its shape, you can change a harmless protein into a potentially deadly one. And none of that is evaluated by our FDA. In fact, by even the biotech companies. This whole proteomics study that found these changes, it was done after the corn was on the market, not by Monsanto, but by independent scientists. Now, in addition to the corn changes, introducing a new allergen, Monsanto's soybeans have a known allergen that is as much as 7 times higher in their cooked genetically modified soy compared to cooked non-GM soy of the same type. Again, collateral damage. Lignin is higher in soy and corn. The metabolic pathway that produces lignin produces rotenone, a plant pesticide linked to Parkinson's. So consuming GMOs might increase our risk of Parkinson's. We don't know because no one has evaluated that risk. Okay, so that's just the process of insertion plus cloning. When they insert the genes and then they blast them into the plate of cells, there's millions of cells there. They don't want to clone them all. So they have to select for which of the cells they're going to clone. So they add, typically, and they've done this for years, antibiotic resistant marker genes into the genetic cassette before they shoot it in. Then they apply antibiotics to the plate of cells, killing nearly all of them except the very few that have the genetic cassette integrated into the DNA of the cell. That means it's working. That means it produces an antibiotic-resistant protein, and that allows them to select which cell to clone because it's the ones that survive the application of a normally deadly antibiotic. Unfortunately, the antibiotic-resistant marker gene remains in every cell of the plant. Now, is there a problem with having an antibiotic-resistant marker gene in our food? Obviously, it might transfer to the DNA of gut bacteria or bacteria in the soil, ending up in a pathogenic bacteria that is now equipped with antibiotic resistance artificially. The British Medical Association was very concerned about it. In fact, the FDA scientists came down strongly against the use of antibiotic-resistant marker genes. They wrote in all capital letters, it would be a serious health hazard to introduce a gene that codes for antibiotic resistance into the intestinal flora of the general population because of the concern that would increase antibiotic-resistant diseases. Now, can genes transfer to gut bacteria? The biotech industry said boldly, no, we don't have to worry about it because genes are destroyed during digestion. Turns out they're not. They're not all destroyed during digestion. In fact, Nature Biotechnology in 2004, in the only human feeding study ever conducted on a commercialized GMO, found that part of the gene inserted into soybeans transferred to the DNA of bacteria living inside human beings. This was in 3 of the 7 volunteers in the UK, and they don't eat a lot of GM soy over there, but 3 of them had the DNA, had the gene inserted into Roundup soybeans that allow the Roundup ready soybeans to not die when sprayed with Roundup. That gene was now inside the DNA of their gut bacteria and may be functioning. We know that gut bacteria was unkillable with Roundup, suggesting but not proving that when you eat a GMO, the genes that are inserted into the GMO might transfer to the DNA of gut bacteria. Is this a unique risk? Is this something that we should be concerned about at all, since we obviously eat food DNA all the time? It turns out that plant DNA does not normally transfer to gut bacteria. Normally, in order to transfer, you need similarity. So this similarity or homology is common among bacteria who pass genes back and forth all the time. But they don't typically pick up genes from plants because they're so different. The genes that we insert into most genetically engineered crops are from bacteria and could have that similarity. If genes did transfer from plants into gut bacteria, they probably would not express proteins because the promoter or on switch, which turns genes on inside plants, doesn't work in bacteria. But the promoter that they use for GMOs is from a virus, and it does work in bacteria. And there's other reasons, the lack of introns, etc., that demonstrate that there are natural barriers that prevent the transfer and expression of plant genes into gut bacteria, which have been disabled with genetic engineering. What this means is that we may be colonizing our gut bacteria with these inserted bacterial genes with viral promoters that might transfer and be taken up by gut bacteria and continue to produce genetically engineered proteins long after we stop eating GMOs. Now, there hasn't been any further research on this, so we don't know, for example, if eating that corn that we talked about, the Bt corn that produces the toxin that breaks open the stomach of insects to kill them, if that would transfer, turning our intestinal flora into living pesticide factories. The biotech industry would say, oh, don't worry about it, the Bt toxin that kills the insects has no effect on human beings. Wrong. It turns out when they did expose the Bt toxin to human cells, it poked holes in human cells just like it does in insects. So we're eating a hole-poking toxin in our corn chips and our cornbread, etc., and that might be bad enough. We might be causing the poking of holes in our digestive tract. It's also a known allergen, so we might be promoting allergic reactions. You can find that in my book, Genetic Roulette, the documented health risks of genetically engineered foods. Well organized. You'll see all the citations. They found the Bt toxin in the blood of 93% of the pregnant women they tested in Canada. So somehow the Bt toxin made it into the bloodstream. Maybe it actually made it in because it poked holes in the walls of the cells along the digestive tract and then got in through those holes. Now, 93% of the pregnant women had the Bt toxin in their blood. The Bt toxin should wash out quickly. Why was it that 93% of the Canadian women who were pregnant had the Bt toxin in their blood. They don't eat corn tortillas every day like they do in Mexico. Most of the corn that they eat there doesn't have the Bt toxin anymore, the high fructose corn syrup, the corn oil. The authors of the study suggested that the Bt toxin may have come from the milk and meat of animals that do eat the Bt corn every day, and that it survived in them and then was consumed by the people in Canada and then ended up in their bloodstream. Is it bad to have Bt toxin in the bloodstream? When it was exposed to the blood of mice, it caused toxicity in their red blood cells. It was also found in 80% of their unborn fetuses. So now you have a hole poking toxin in the blood of an unborn fetus that doesn't have a blood-brain barrier. The hole poking toxin may end up damaging the brain. We don't know. Again, these are some of the studies that need to be done. We can't simply ignore these possibilities, especially when there's such a tight correlation now, not proving, but correlation between so many diseases on the rise in the US and the use of GMOs, including autism, diabetes, certain cancers, all sorts of gastrointestinal disorders, deaths from stroke and Alzheimer's and dementia, etc. Some of these may be related, but no one is looking. Now, you have the process of transfer of genetic engineered material in from one species to another. Just that process, you're crossing the species barrier. Nobel Prize winner George Wald said, this is not to be confused with traditional breeding. Listen carefully, Neil. Not to be confused with traditional breeding. It is transferring genes between totally unrelated species. Now, what might happen in that case? Australians were developing peas to produce an insecticide. And they took a gene from beans and put it into the peas to kill a pea weevil. They tested the peas like normal GMOs are tested, and they passed with flying colors and were ready to go on the market. But these developers decided to do one more test. They decided to test the immune system response from mice who were exposed to the peas versus who were exposed to the natural occurring protein found in the kidney beans. So they took the normal kidney beans, exposed it to mice, no problem. They took the exact same protein, although this time it was transferred into peas, exposed to mice. The mice had 5 inflammatory reactions, suggesting that the peas had become potentially deadly. So they canceled the production of these genetically modified peas. They believe that the reason why this otherwise identical protein had become dangerous, potentially deadly, is that once it transferred into the new environment, sugar molecules attached themselves to the protein with a different shape, a very subtle difference. This is called glycosylation. It's known to promote allergic reactions. They don't even check with the current GMOs on the market whether there's glycosylation changes. They just assume that there isn't. This group happened to use a research technique that almost no one looked at for the current GMOs to find that difference in the sugar patterns. And they were surprised that when they transferred genes between such closely related species as beans and peas, that there was such a dramatic difference, turning a harmless protein into a potentially deadly one. But Monsanto and the biotech industry transferred genes not from closely related species, but from different kingdoms, and just assume that they will not have the added molecular attachments, assume that they'll be folded correctly, assume that they'll be produced correctly. All of these assumptions have not been validated. Now, in addition to the Bt toxin built into our corn and our cotton, most of the GMOs are engineered to withstand doses of Monsanto's Roundup herbicide. If you have been current in the research on Roundup, you'll know that it is now linked to birth defects, Parkinson's, endocrine disruption, all sorts of problems. There was a review study that linked it to heart disease, cancer, obesity, diabetes, multiple sclerosis, anorexia nervosa, digestive disorders, reproductive disorders, another one, gluten sensitivity, kidney damage, aggression, depression. If you look at the nature of Roundup, it's too big of a discussion here. It is not the benign chemical that Monsanto has been thrusting upon us. It is described by many scientists as the most chronically toxic chemical on the planet because of how much it's used and how insidious it is in breaking down certain metabolic pathways of our gut bacteria, of our detoxification, chelating with minerals, causing endocrine disruption, promoting the growth of cancer cells, etc., etc. So we now have a situation where these untested GMOs are on the market. When the American Academy of Environmental Medicine reviewed the animal feeding studies 5 years ago, they said there's enough evidence to recommend that all physicians in America prescribe non-GMO diets to every patient. Thousands of doctors are doing just that. And they report to me at our Institute for Responsible Technology, as do the people who've switched from GM to non-GM, that they're getting better from the same type of diseases and disorders that were found in the lab animals that were fed GMOs. Gastrointestinal problems, immune system problems, organ damage, dysfunctional regulation of cholesterol and insulin, reproductive disorders, accelerated aging. These are the things described by the American Academy of Environmental Medicine, and they're among the categories that we hear by thousands of people who are switching to non-GMO diets. This is anecdotal. This is not hard science. But if GMOs are causing these problems, it's supportive evidence, just like the correlational charts showing an increase of GMOs paralleling the increased expansion of these diseases and disorders in the United States. When livestock are taken off of GMOs, unlike when humans switch, when they have to go organic or reduce processed foods, introducing all sorts of confounding factors that might cause the interpretation of why they're getting better, makes it very, very hard to identify. But when livestock are switched from GM to non-GM soy or corn, there are no confounding factors. It is simply a single change. And the farmers and veterinarians tell us over and over again that the animals within days are using less medicine, their behavior is different, their death rate is down. It's very dramatic, and it parallels, same with pets, it parallels the problems found in the animal feeding studies, getting better in humans, getting better in livestock, getting better in pets, on the rise in the U.S. population since GMOs were introduced, and those same diseases and disorders would be predicted by looking just at the nature of the two main toxins in GMOs, the Bt toxin and Roundup, which is sprayed onto most crops, or even looking just at the massive collateral damage that occurs as a result of the process of genetic engineering. That could explain a wide variety of problems. So we don't have to create a clear causative relationship in this argument for Neil deGrasse Tyson. We don't have to say it's been proven that GMOs are linked to autism or whatever. We just have to show that the mechanics of the process of creating genetically engineered crops and how they're grown and sprayed and what they produce is so different from traditional breeding that you cannot hide behind that cloak and pretend that simply because we have been altering genetic structures through selective breeding for centuries, we can ignore these bacterial genes with viral promoters inserted using biolistics into crop cells, cloning them into crops, which creates all sorts of extra mutations, and then ignoring all of those changes on the protein level, the RNA level, the metabolite level, and then feeding them to an entire population with no evaluation, no proper epidemiological studies, and just going ahead with the plans of the biotech industry to genetically engineer 100% of all commercial seeds. This was their ideal future, Monsanto's ideal future, described by their consultant in a January 1999 conference. So they want to replace the products of the billions of years of evolution with designer organisms using designer genes designed for greater profit and control. That is their ideal future, which has only gotten expanded beyond seeds to now introduce insects and livestock and trees, etc. Before we do this, we need to answer those deep questions, look at the science, to quote one UC Berkeley professor, put it back in the laboratory for 50 to 100 years until you've done your homework. But patents expire, return on investment demands in the marketplace require faster action. So this technology, which I'm not against generically, but in its current form, it is so dangerous, it is so immature, but it has been thrust into the food supply and put out into the environment where you cannot recall genetic contamination long before the science was ready. So Neil deGrasse Tyson, get with the numbers. If you don't understand what I've said, if you haven't absorbed it, if we want to spend some time, let's get together for a few hours and discuss it. Or better yet, roll a camera, we'll debate with some time to go into these principles, and you'll see that this basic, simple excuse that we've been modifying genes for years through selective breeding, so we don't have to worry about GMOs, it doesn't hold water. Safe eating, Neil, and everyone else.